So the last couple of times we've been talking about examples of where the immune system is depressed. And so we spent some time talking about microbial mechanisms that basically try to dampen the immune response such that the immune system will not eliminate those pathogens. And it's part of their evolutionary strategy to try to avoid immune detection. We also talked about genetic diseases where, as a result of either dominant or recessive mutations in certain key genes that are involved in immune responses, the result of that is that also leads to defects in immune responses. And for the next 3 lectures, we're going to actually talk about examples of where the immune system has exactly the opposite problem, and that is that the immune system is essentially hyper-responsive. And in the first 2 lectures, today and then again on Monday, we'll talk about some examples of what we call hypersensitivity responses, where basically the immune system is hyper-responsive to antigens or what we call allergens that come from the outside. And then we'll follow that up with a lecture following Wednesday, the day before Thanksgiving, where we talk about autoimmune disease and how the immune system can recognize our own tissues if there's a problem with self-recognition, or I should say self-tolerance. So today we'll talk about this concept that there are basically 4 types of hypersensitivity responses that are driven by primarily T cells and B cells in this case, and we'll talk about some examples of the collaboration between T cells and B cells that give rise to that hyperreactivity. And today we're really focused primarily on type 1 hypersensitivity and some examples of that with respect to what you might see. We'll talk about the involvement of mast cells and how they become activated in response to IgE binding, and that results in the release of a bunch of inflammatory mediators, including histamines, that also leads to the activation of a cell type, a granulocyte cell type called eosinophils that are also involved in these kinds of IgE-mediated allergic responses. And then we'll talk about systemic anaphylaxis, allergic rhinitis, and then finally asthma. So before we start, I want to get a sense from the group here today, and maybe the group here that's not today, but which is a substantial number of you guys. I don't know where you are, but I guess we'll meet you at the final exam. But thank you guys for all coming. So of the group that's here, how many of you guys have some sort of an allergy? I mean, that's a pretty substantial number. That's probably about half of you, something like that, that are allergic to something. So maybe throw out there, not to get too personal or anything like that. So if you don't mind, what are you guys allergic to? And don't say your professor in M121. All right, yeah, over here. Penicillin. Okay, so we'll talk about that. We'll spend more time talking about that the next time, where we talk about type 2 hypersensitivity. Back there. Peaches, really?
Yeah, I got it a couple years ago.
Huh.
I found out through banana.
Okay, so no peaches for you. Okay. Over. To what?
Sunlight.
Is it possible to be allergic to sunlight? Well, people have sensitivities to sunlight, but it's not in sort of the same kind of hypersensitivity responses that we'll be talking about. So you can have hypersensitivity to that. A lot of people easily get sunburns. People that have certain types of mutations in DNA repair pathways, for example, are hypersensitive, but it's not an immunologic basis for it. Some other examples. Sorry, yeah. Yeah, so you get hay fever, that sort of thing. So you're hypersensitive to pollen, that kind of stuff. Yeah, exactly. I am too, actually. Especially when we have for me, it's when the Santa Ana winds come in and all that junk. How about food allergies? So we had one over here with peaches, yeah?
Peanuts and soy.
So glutens and soy, okay.
Peanuts.
Oh, peanuts, yeah, absolutely. So I hope you have your EpiPen. Good, okay. So, and we'll talk a little bit about epinephrine towards the end when we talk about anaphylaxis. So obviously there's a lot of allergy that's out there. And it's kind of weird, you think, almost half the people in this room have some sort of allergy. What's going on? And actually, if you look This is something that's fairly recent. So we know that people have become much more susceptible over the last 30 or 40 years to allergies. So why is that? What do you guys think? And we'll get into some of the hardcore stuff in a bit, but this is kind of an interesting thought experiment because we actually don't know the answer to this, but what do you guys think might be the reason for it? Any thoughts? Yeah. So we live in a clean environment. So this is part of what they call the hygiene hypothesis. And we'll talk about that more when we talk about autoimmunity, but it's this idea that, you know, if your immune system isn't busy fighting off, you know, worms and viruses and all that kind of stuff, because it's so clean, then it gets, you know, bored. And just like a teenager does bad things. So that's one possibility, and there's definitely some data to support that may be the case. Any other thoughts? Yeah, over here.
Dealing with like the westernized diet and how processed our food is.
Processed food, right. So our Kraft macaroni and cheese is so clean that it doesn't have the antigens that would keep, again, sort of keeping our immune system happy. And maybe it's introducing new things into the body that might be promoting some sort of an immune response or something like that. So I think those are all, I think, reasonable ideas. There's also this hypothesis, especially with respect to asthma, that because we're living in, you know, places like this, basically, that have, you know, no open windows and no open doors, that the result is that you sort of trap in these allergens that are constantly being recycled through this, you know, heating and ventilation systems, and that tends to accentuate our exposure to some of these allergens, and then ultimately we become hypersensitive to those. So I thought we would talk a little bit about this first because I don't really go into the details in the chapter, and your book doesn't really go into the details of this either, but it's an important thing to consider. So clearly there are environmental issues. We also know that there are certain inherited issues as well. So there's probably a genetic basis for it. You tend to find that in some cases, some of these hypersensitivities tend to cluster. There's a genetic basis for it as well. And we'll talk about some examples of that in particular next time. All right, so let's talk first about the allergens. So we just spent a little bit of time talking about some of the different allergens that people are sensitive to here in this room. And some examples of that, somebody, you know, has hay fever, and that's a result of plant pollens typically, or other things that are present in the dust. A lot of people have sensitivity to dander from domesticated animals. My wife is hypersensitive to cat dander, despite the fact that we have a cat, which doesn't make sense to me. But in any event, you know, a lot of people have that hypersensitivity. It's also true for mold spores, et cetera. And other people are sensitive to insect venoms. So I think we had one person hypersensitive to bee venom. Anybody else here? If you get a bee sting, you got to get your EpiPen out. Yeah. So my nephew has a hypersensitivity to bee venom. So if he gets stung, he could basically undergo anaphylactic shock and die if he doesn't quickly deal with that. And so he needs to have an EpiPen, which we'll talk about. Other examples, vaccines. Some people are hypersensitive to drugs. You know, penicillin, we heard somebody talk about that. So there's a lot of antibiotic hypersensitivity that's a problem. And other kinds of drugs as well. Other people have sensitivities to, in particular, food allergens. So, you know, one example of that is peanuts. We heard somebody is hypersensitive to peanuts. Some people are hypersensitive to shellfish. One of our friends, their daughter is super cute. She's probably about 4 years old. She's hypersensitive to just about everything. So her mom, anytime they go to a restaurant, I feel horrible because, you know, first of all, I feel bad for the chef because they have essentially nothing they can provide this kid. And she has to, you know, the mom has to bring around, you know, these special, you know, food items that she knows this kid isn't hypersensitive to. And again, these are things that, you know, it's not just a matter of, you know, maybe getting a little rash or something. This is something that could actually be really very serious and could put that little kid in the hospital. And then there's also examples of contact hypersensitivity where, you know, for example, if you touch poison ivy, or some people have hypersensitivities to nickel and other metals, and that sort of contact hypersensitivity we'll talk more about the next time. So all these different types of allergens that are out there, for the most part, these are relatively innocuous. They're clearly not things that are going to kill us. So why is it that we're hypersensitive to these things? And we'll talk a bit about that today. All right, so the first thing I wanted to go over is just the different types. And like I said, today we're really going to focus on this type 1. We'll spend more time talking about the type 2 through 4 the next time. But the ones that we'll really focus on today are for example, allergies, which is basically where the immune system responds to innocuous antigens from the outside. And we call these hypersensitivity responses. And basically the kinds of different hypersensitivity responses that are out there are really based on, the reason why we classify them is really sort of based on their, the type of antigen and the immune response, the mechanism that's at play. And in this case, type 1 through 3 are primarily humoral, so they're involving mostly antibodies, whereas type 4 is a T-cell dependent. So type 1, which is also shown over here, this we know is the result of a specialized antibody isotype called IgE. So IgE, if it recognizes some of these food allergens or other allergens, the result is that, which are soluble antigens that make their way into the mucosa typically. So they come into, into the gut through an oral route, or they come into your lungs because you're breathing this stuff. The result of that is that if you have these IgE antibodies, they can then bind to that allergen. That then leads to binding and activation of what are called mast cells, which are essentially an innate leukocyte that's part of our innate immune system. And the result of that is that you then can develop hypersensitivity where these mast cells get activated, and I'll go over the details of that in a bit. But some examples of this that we'll talk about are allergic rhinitis, asthma, and then anaphylaxis. Type 2, as we'll hear about, these are against small molecules. These are mediated by IgG isotype antibodies. And these are usually antigens that are cell or extracellular matrix associated proteins. And in this case, cells that have this Fc receptor, in this case a gamma receptor, are going to bind to these IgG antibodies that are specific for the allergen. That will then lead to activation of these mast cells, and the result of that then is that you get a response that would be inappropriate. And these are typically in response to things like penicillin. So, if some of these antibiotic sensitivities are because of the fact that these are fairly reactive proteins, as we'll talk about the next time. And when they interact with some of these membranes on cells, they can generate what we call neoepitopes or new epitopes. And then the consequence of that is that you can then get complement activation and then destruction of the cells. And this is an example here where you have platelets, which are essentially little pieces of megakaryocytes that are involved in basically in blood clotting and wound repair. Those then get targeted for destruction through this phagocytosis, say into a macrophage or what have you. And then the type 3, which are due to what are called immune complexes. This is where the antigen is basically bound by multiple antibody molecules, and that results in deposition into the walls of small blood vessels or into the lung, the alveoli in the lung. And again, this is mediated by an Fc receptor. Result of that is that through this deposition of these immune complexes, you get localized tissue damage, and that can cause some problems down the road. And we'll really focus on these guys the next time. So I want to focus on this type 1 mechanism here, and we'll talk about how that's mediated. And then finally, the type 4 reaction, which, as I mentioned, it's cell-based. So it involves T cells, primarily the Th1 cells. It's also called delayed type hypersensitivity, or DTH. And DTH is typically a problem, for example, with, if you have a response to certain insect venoms, poison ivy, and so forth. And so the consequence of that is that you get this response of T cells, primarily Th1 cells, and so the consequence then is that it takes some time. So the reason why that's called delayed type hypersensitivity, as opposed to some of these acute hypersensitivity responses, is that it takes somewhere between 1 to 3 days for it to take place. That's time where you have these antigen-reactive T cells getting activated and then differentiating primarily into the Th1 cells, although there is a component of Th2 cells involved in this as well. And this is typically in response to certain soluble antigens that, for example, can lead to the activation of macrophages through Th1 cytokines. And that results in these macrophages then basically releasing chemokines, cytokines, and cytotoxins that can then lead to localized tissue inflammation and damage. You can also get activation of Th2 subsets, which will result in the activation of eosinophils. And we'll talk a little bit about these cells today. They're part of what are called granulocyte, the granulocyte subset. And they're involved in things like asthma and so forth. And then you can also get activation of cytotoxic T cells, typically in response to cell-associated antigens, and that can lead to localized cytotoxicity. So you get localized damage, and an example of that is with contact dermatitis. So we'll spend more time talking about this the next time. Okay, so the first thing that I want to talk about are these mast cells and how they are involved in mediating some of these allergic responses. So as I mentioned, these allergies are basically these type 1 responses, and they depend on IgE. So IgE needs to be produced. It needs to be produced in such a way that it's specific for what would essentially be an innocuous antigen, but somehow that antigen makes its way into some mucosal site in the body, either through the lungs or through the airways or through the gut or through the skin. And then the consequence is you activate both T cells and B cells. And in this case, the T cells that are important in this case are Th2 cells. And so the consequence of that is that you can get activation of these mast cells. And so how does that work? So basically, let's say that you have some IgE that's been produced. You've been sensitized at some point in your life to, pollen, for example. The pollen comes in because you're breathing during some Santa Ana wind condition. And so the consequence then is that this pollen makes its way into your airways and then ultimately makes its way inside of the lamina propria of that mucosal site within the lungs. So the consequence of that is that if you have this IgE specific for some of these pollen antigens, it can then bind, this IgE bound to the antigen, can then bind to a mast cell. And on the surface of a mast cell, there is a special Fc receptor. And you guys have heard about Fc receptors before. We talked about that in the context of, for example, naive B cells and how certain naive B cells, because they express these Fc receptors on the surface, that blocks naive B cell activation. But in this case, this is a special Fc receptor that's specific for IgE. So it's an Fc epsilon receptor 1, which is basically made up of 3 different chains: an alpha chain, a beta chain, and this gamma chain dimer. So the result of this binding of this IgE bound to an allergen is that activates this receptor complex, and then that leads to what's called mast cell degranulation. So these mast cells they're very dense with these granules, just like we were talking about with cytotoxic T cells that have these cytotoxic granules. But in this case, these granules have a whole bunch of different components, which we'll talk about in a couple of minutes. But basically what those granules do is that once this mast cell is activated by these IgE molecules, that then causes this release of these granules, and then you get this localized immune response. So I wanted to show, and this is just a figure basically showing what these mast cells look like with these dense granules that are basically staining here in red. So you can see these little circles inside of this mast cell. Those are filled up with all sorts of pro-inflammatory mediators that can then become activated. Now, this response, normally you would think, well, why would the immune system even make IgE if it's such a bad thing and all of us have these allergies? Well, it's primarily an anti-parasite response. So we were talking before about some of these intracellular parasites like toxoplasma, and so forth. And basically what IgE is really good at is bringing a response against some of these parasites. So, and in addition to that, it also, these kinds of responses against these parasites involve these granulocytes, both what are called basophils and eosinophils. And the mast cells, when they degranulate, in addition to releasing factors that can basically affect the local tissue and cause control of that parasite, they also bring in these granulocytes, basophils and eosinophils, which can then deal with that localized infection. So we think of an allergic response as basically being an anti-parasite response that's kind of out of whack. So the immune system is not responding to parasites, but instead is responding to these allergens.
Some people develop an allergic reaction or hypersensitivity when exposed to substances such as dust, pollens, animal dander, or penicillin. This hypersensitivity is mediated by IgE. Sensitization occurs when the antigen makes contact with some part of the body. The antigen is taken up, processed by antigen-presenting cells, and presented on a class II MHC to T helper cells. Tissues under the mucous membranes are rich in B cells committed to IgE production, and IgE-producing cells are more abundant in persons susceptible to allergies. The T helper cells produce cytokines which stimulate these B cells to proliferate and differentiate into IgE-producing plasma cells. As IgE is produced in specific areas of the body, the IgE molecules attach via their constant regions to receptors on nearby mast cells. Mast cells contain granules packed with chemicals that induce a hypersensitivity response. Once attached, the IgE molecules can survive for many weeks. The individual is now sensitized to the antigen. When exposed to the antigen for a second time, the antigen binds to the IgE antibodies on the mast cells. To trigger a response, two cell-bound IgE molecules must react with a specific antigen. Within seconds of the reaction, the mast cell releases histamine and other mediators of the inflammatory response from the granules, triggering a variety of symptoms.
All right, so I just wanted to show really quickly what the degranulation looks like. So these are some mast cells here, and you can start to see they start to very quickly release these little granules out into the outside of the cell a lot, and we'll talk about what some of the components are there. But importantly, those granules are then going to be able to mediate this anti-parasite response, or at least that would be the normal case. But in allergy, they then lead to some significant changes in the local cellular, the local physiology of that tissue. All right, so some of the factors that are present in these mast cells, these guys here in red, those are the ones that actually exist in the granules. And some examples of those, there are certain enzymes that are involved in remodeling of the tissue matrix. So they basically are trying to modify the matrix and modify that connective tissue to allow the appropriate immune response to take place. Some examples are tryptase, chymase, cathepsin G, et cetera. Certain toxic mediators like histamine and heparin that are toxic to parasites can increase vascular permeability in an attempt to try to wash that parasite out of that tissue through the blood, and then also induce this smooth muscle contraction. In addition, there's also cytokines like TNF, which is also thought to exist in these granules, that it's a pro-inflammatory cytokine, so it mediates the recruitment of inflammatory cells, and the result of that is that you would then try to eliminate this parasite. There's also other cytokines like interleukin-4 and 13 that are involved in promoting the Th2 response. So you differentiate to Th2, you get T cells that can then help B cells to make these anti-allergen antibodies or anti-parasite antibodies, and then the result of that is that they then start to produce IgE. And other cytokines that are involved in production and activation of what are called these eosinophils, these specialized granulocytes that are activated by these mast cells. Also chemokines that are chemotactic for some of these inflammatory cells like macrophages and neutrophils. And then lipid mediators that have a really important role in controlling the local tissue remodeling process. So for example, this also leads to smooth muscle contraction, increase in vascular permeability, and an increase in mucus secretion. We'll talk about how those are all playing a role. and then also platelet activating factors that can activate, are basically chemotactic for some of these leukocytes, and then can activate some of the production of some of these leukotrienes, which are promoting some of this response. All right, so how do we get the IgE in the first place? I mean, what are the allergens, and how does somebody become hypersensitive to this? So first off, some of the features of these antigens, and in this case we're talking about inhaled antigens, but this is somewhat similar in other cases. So let's say that here you have somebody that's exposed to pollen because it's blowing through the air, makes its way into the airway, and so the result of that then it gets brought into the airway. It can start to make its way inside of this mucosal site where you have some antigen-presenting cells that can then present that antigen to a T cell. And so the consequence then is the T cell becomes activated, it meets up with the B cell, and that B cell, one thing that the T cell is doing is providing help through CD40 ligand. It's also producing interleukin-4 because it's a Th2, and that the combination of those two signals then cause this B cell to switch, to undergo this isotype switch to make IgE. So you now have IgE that's then specific for this particular allergen, and then that IgE that's then produced by, in this case, a plasma cell, can then go on and bind to these Fc epsilon receptors on a mast cell. And so the consequence of that then is that you get this allergic response. So some of the kinds of antigens that drive this, that act as good allergens, are, for example, proteins, because proteins are required to induce a T cell response. They're typically enzymatically active, and an example of this are proteases, and I'll show you on the next slide why that's usually the case. They're low dose, and so we talked a little bit about what conditions drive a Th1 versus Th2 response, for example. But one of the things that I didn't mention is that the dose of the antigen, the amount of signal that comes through the T cell receptor, can dictate whether you're going to get a particular helper cell differentiation. So Th1 cells require a lot more antigen than do Th2 cells. And so in this case, there's not a huge amount of antigen, and so that tends to favor the generation of these Th2 cells that produce IL-4. It can also, the allergen needs to be able to basically come out of the particles, in this case, if it's pollen, and make its way inside of the mucus so that it can ultimately make its way inside of this, the lamina propria of this mucosal site. And it needs to be readily eluded from the particle. It needs to be able to get out, and it needs to survive as a desiccated particle, at least in this case, if it's going to come by way of the air, and obviously needs to be required for T cell priming. So in this case, it needs to be able to bind to MHC class 2, or some of those peptides need to get into class 2 to be able to activate a T cell response. So an example of these, I mean, we talked about pollen. Another example are these dust mite allergens that can lead to, for example, asthma. And there are specific molecules that tend to be particularly good as allergens that are proteolytic enzymes. And one of these is called DIR-P1. It's basically named after the dust mite itself. And this dust mite, it releases anyway, so it poops on things. And this particular allergen comes from that. And what it does is once that gets in the airway through the air, this DIR-P1 as a protease can cleave these gap junctions. So this is the airway epithelium, and in order for this allergen to make its way inside of this mucosal site, it needs to basically get past these gap junctions. So that barrier that's normally there is broken down because this DIR-P1 protease can then cleave this gap junction protein called occludin. And so the consequence of that then is that this epithelial layer is now permeable. The allergen can get inside this cell, or inside of this tissue, where it can then bind to an antigen-presenting cell, lead to T cell activation, lead to the production of IgE, and then the degranulation of these. Okay. Any question about these dust mite allergens? All right. So as I mentioned, this process is dependent on IgE, which is capable of binding to these mast cell IgE receptors or Fc receptors. And so there are basically two reasons or two components to this that promote this IgE switching. So one of these is that these allergens are typically involved in the switch to Th2 cells, and then the cytokines and costimulatory signals that are presented to the B cells will then produce this switch to this Ig epsilon locus. And then, as I mentioned, the antigen, can enter various ways. If it comes in through the skin, there are certain components in the immune system that are then particularly geared for a Th2 response, resulting in this IL-4. So the consequence then is basically two different ways in which the Th2 response can evolve. So the first shows a specialized type of T cell that we call NK T cells. And these NK T cells have on their surface a special subset of T cell receptors that can bind to CD1. And we talked a little bit about CD1 in the past with respect to different types of antigens, the non-peptide antigens that they can present. But on these NK T cells, we know that they can participate in this sensitization. And an example of that is that an NK T cell can then become activated, start to increase the production of IL-4, and the result of that then is that if you have any naive T cells, standard T cells around, they, in the presence of this IL-4, are then activated by the allergen and then tend to switch to this Th2 phenotype. So, the one way that we talked about is that with low dose of this antigen that's present, you tend to favor Th2, but you can also, because of the fact that you have these NK T cells around, because those cells are producing IL-4, that can also tend to favor the generation of these Th2 cells. And again, the Th2 cells, by producing IL-4, passing that along to a B cell, you then get isotype switch and plasma cell generation such that you get a lot of IgE produced. All right, so then what are the effector mechanisms? Once the IgE is secreted by these B cells, these plasma cells, and it's present in the body, then how does that activate? What's the consequence of that with respect to the mast cells? And so they're basically different mechanisms in these mast cells, and we talked about some of the different compounds, the different effector molecules in these granules. But one thing that's really important to note is that where this is in the body, where these mast cells are getting activated, is really going to have a very important role in the sort of clinical outcome. So for example, if their mast cells are present in the GI tract, this can then, once they become degranulated, this can then result in increased secretion of fluids, increased peristalsis, and then the idea behind this is that if this was a parasite, you want to try to get rid of it. If it's in the gut, you activate this gut immune response, and that's going to then lead to either diarrhea or vomiting. So from one direction or the other, or from both, you're going to get rid of this bug. Okay, but obviously this is something that's inappropriately activated in this allergic response. If it gets in the airways, as we talked about, you're going to get localized activation of mast cells. The result of that is that some of these inflammatory mediators can decrease the diameter of the airway, and so the consequence, and then you also have an increase in secretion of mucus, which basically makes you start to wheeze, you start to cough. There's a lot of phlegm, and so the purpose of this is to try to basically eliminate that bug from your airways, and obviously this makes you feel kind of sick. And then in the blood vessels, this can then lead to increased blood flow and increased permeability of the blood vessels. The point of that is that the blood is essentially trying to wash some of these parasites out of the system. And as we'll talk about in a little bit, that's kind of a problem in terms of anaphylaxis. Okay, but again, the key is that you need to have these mast cells, and they're getting activated by the allergen cross-linking the IgE antibodies. Okay. So some of the different types of allergic responses that are mediated by IgE include anaphylaxis, and this is in response to drugs, serum products, venom, peanuts, and this is typically something that somehow makes its way intravenously, either directly or through absorption into the gut. And the consequence is that you have edema, which is where you have a lot of fluid retention, increased vascular permeability, the windpipe becomes occluded, and then you can have circulatory collapses we'll talk about, and then death. And a treatment for this is this epinephrine, which is also known as adrenaline. So we'll talk about in a minute why that works. It's also wheal and flare, basically insect bites, allergy testing, and I'll show you an example of that in a minute. This is where the the allergen enters subcutaneously, so it comes in under the skin. And this causes localized swelling because of an increased blood flow and vascular permeability. Allergic rhinitis or hay fever, this is obviously coming in through the, through our breathing. And examples of this would be pollen and dust mite that can then lead to this localized edema, in this case in the nose, which is why they call it rhinitis. Also bronchial asthma, again from these similar kinds of airborne pollen and dust mite feces that can lead to bronchial constriction, misproduction, et cetera. And then food allergies that obviously come in through our gut, through our mouth actually. And examples of that are shellfish, milk, eggs, fish, wheat, et cetera. And that can then result in problems obviously in our gastrointestinal tract with vomiting and diarrhea, but also leads to other things like itching, hives, and anaphylaxis. All right, so I said that I would show you an example of one of these hypersensitivity responses that you might go to an allergist and find out, well, what are you hypersensitive to? So they may do something called a skin test where they take a little bit of that allergen and basically rub it on your skin and then look to see whether you get a response. And so there's actually two different responses that you can see on this sort of skin test, an immediate response and then what's called the late phase response. That's also true for asthma. So this is a skin test, but as a physician, one might also want to look at certain allergens that can then induce asthma, allergic asthma. And you also see a very similar kind of response in these type 1 hypersensitivity responses where you have an immediate response and then a late phase. So here, obviously, the immediate phase is you get this little bump, and then the late phase, you have this much greater swelling that's going on in that tissue. The reason for that is that for the immediate phase, presumably here you have pre-existing IgE antibodies to this particular allergen. So you've been sensitized at some point in your life to that allergen, and you have those IgE antibodies. And so then you get this increase in histamine from the mast cells, which then leads to this localized increase in vascular permeability and airway smooth muscles, at least in this inhaled response. And so then, obviously, these mast cells that are binding to the IgE are really important in this. So you need to have the pre-existing IgE, but it's relatively short-lived, and so this is pretty small and pretty localized. In the asthma model, you can see that there's this. So this is a measure here of expiratory flow, which is basically your breathing rate. So the ability to take in air is pretty significantly reduced. So you would go, it's almost 8-fold reduction in the amount of air that's being taken up as a result of this response. But that then sort of resolves itself within about an hour, and you seem to be breathing essentially normally. But then several hours later, you start to have this secondary response, this late phase response, where the breathing starts to become labored again. And the reason for that is that at this point, now you have a whole bunch of cells that have gotten recruited by these mast cells. The mast cells degranulated, they release these inflammatory mediators and chemokines, and the result of that now is that you have these leukocytes that are coming in and then inducing this late phase response. And so you need to have Th2 cells in order for this to take place. And some of the mediators that are produced by these mast cells include these leukotrienes, which I mentioned earlier, which are then basically leukotrienes and then converted into these what are called prostaglandins, which are pro-inflammatory. All right, so I already mentioned this point that it really depends on where these mast cells are getting activated. That depends on that dictates the sort of response that you're going to get, the clinical response. But the other thing that's important is the amount of antigen, specific IgE. So, have you been sensitized before? Oftentimes your first exposure isn't particularly problematic, but then on subsequent exposure, this is when things start to get much more serious. The route of administration, as I mentioned, these different sites where the mast cells are getting, and then of course the dose of the allergen itself. So if you're exposed to a very, very small amount, unless you're really hypersensitive, you're not going to get particularly sick. You might have some hives or something like that. So all three of those are important to the allergic response. All right, so I want to talk a little bit about anaphylaxis. And basically anaphylaxis, as I mentioned, is a situation where the immune system, because of this hyperreactivity, you get all sorts of things happening that can be pretty bad. So I wanted to talk a little bit about what an example of this is. So for example, let's say that you're hypersensitive to penicillin. This then comes into the bloodstream, and if it's coming in through the bloodstream, it can be disseminated throughout the body. So this could then lead to a systemic hypersensitivity response. And just an example of that here is you have these blood capillaries. Penicillin's going through and then ultimately makes its way inside of the tissue, where you then have these pre-coated mast cells with IgE on the surface that can bind to this allergen, and the result then is that you get the degranulation of these mast cells. In the heart, again, because it's in the blood, you get this increased permeability of the capillaries and entry of fluid into the tissues, swelling of the tissues, including the tongue. This can then ultimately result in loss of blood pressure, and obviously associated with that, you have reduced oxygen being brought to the tissues, which can then lead to irregular heartbeat, heart failure, and then anaphylactic shock, and then you can actually become unconscious and worse. And then in the respiratory tract, again, because of some of these leukotrienes, the result is that you have this smooth muscle contraction, and the result of that then is that you have difficulty with constriction of the throat and the airways, so you have difficulty in swallowing and difficulty in breathing. And in the gastrointestinal tract, contraction of the smooth muscle then can lead to stomach cramps, vomiting, and then increased fluid outflow then can give rise to that stuff. All right. I just can't say it when it's lunchtime, sorry. So what is the treatment? What would you do? If somebody comes in, I mean, we talked a little bit about this EpiPen. So if you're hypersensitive to bee stings, for example, you need to have an EpiPen around so that you can treat it. So basically what epinephrine is it's a vasoconstrictive drug that can also induce tight junction reformation. So as I mentioned, the vasculature is quite leaky as a result of these mast cell inflammatory mediators. In this case, the epinephrine is going to actually cause that to then become vasoconstrictive. This also leads to tight junction reformation, and then it can relax some of these smooth muscles in the airway, so now you can start to breathe. And so the result of that then is that you can control your irregular heartbeat and this reduction of oxygen that's getting to the tissues. You can basically deal with that. So this then allows you to overcome this anaphylactic shock. There are other ways that you can treat this, including antihistamines, which can block some of these inflammatory mediators produced by the mast cells, as well as corticosteroids, but these are the main, epinephrine is the main approach. And I just want to show a quick movie on that.
Anaphylaxis is a systemic reaction that can occur when a patient who is allergic comes into contact with the allergen they have been sensitized to. Clinical symptoms may be mild, moderate, severe, protracted, or biphasic. The allergic response begins with exposure. As antigen molecules enter the bloodstream, they interact with IgE antibodies bound to mast cells and basophils. This union of antigens and antibodies produces a complex sequence of intracellular biochemical events in the form of a cascade. A signal sent through the IgE antibody to the cell's interior begins the sequential activation of a series of enzymes. This leads to the influx of calcium into the cell and the fusion of granules with the cell's external membrane. The result is degranulation, with the release of histamine and other chemical mediators into the extracellular space. These mediators flow through the body and bind to receptor sites at target organs, producing physiologic effects such as vasodilation and increased vascular permeability. In the lungs, histamine produces smooth muscle contraction, which causes obstruction, wheezing, and shortness of breath. A combination of mechanisms results in myocardial depression. To overcome these life-threatening events, epinephrine must be self-administered without delay. Upon injection, epinephrine works rapidly at organ sites to counter the effects of chemical mediators, acting to constrict blood vessels and reduce vascular permeability, relax smooth muscles of the airway, and stimulate the heart rate. This generally reverses the most dangerous effects of an anaphylactic reaction, allowing time for transport to an emergency center for full professional care. However, in some cases...
All right, we don't want to hear about the other cases. All right, so the last thing I want to talk about is rhinitis and asthma. And I mentioned earlier that rhinitis is a situation where you have a localized inflammation in the nose and in the upper airway tract that is caused by some of these allergens, and a good example of this would be hay fever. And so the histamine that's then being produced by these mast cells can then lead to the recruitment of another cell type that are called these eosinophils. And these are again, granulocytes. They can make their way inside of the mucosal site, and the result of that is that they become activated. You start to get some of these, they themselves produce some of these inflammatory mediators, and then you get this localized inflammation, which can then lead to enhanced production of mucus, as well as difficulty breathing. And so you get a runny nose and so forth. So with asthma, the situation can be a little bit more serious because of the fact that there's an acute response, and then later there's a chronic response. So the acute response is very similar to what we were talking about. You have entry of the allergen through the airway into this mucosal site, activation of mast cells, and then this leads to then disruption in smooth muscle and so forth. And so now you have contraction of the smooth muscle, increased secretion of mucus, so very similar to the situation with rhinitis. However, there's a later stage. So if this is happening over time, the result is that you start to recruit in some Th2 cells, and obviously that's going to help with greater production of IgE. But on top of that, because they're producing Th2 cytokines, the result of that is that these can then lead to enhanced tissue damage, enhanced recruitment of eosinophils, and then over time you get pretty significant tissue damage. And so examples of that are shown here. So this is a person who's had this chronic reactivation of the immune system in response to these kinds of allergens coming through their respiratory tract. And so now you start to see this pretty significant occlusion of the airway. So this is the bronchus, and this particular patient died because this is all they had. This is the only space that's present in their airway to be able to breathe. And so the result of that is that this person, by chronically re-stimulating this kind of allergic response, it ultimately led to death because they just couldn't be able to breathe very, very efficiently. So this person was probably had to receive some sort of an intubation and oxygen and all that, but ultimately that apparently failed. All right, so we'll finish it there. We'll come back again on Monday, so don't forget that on Friday, Ryan will hold a review session here, and then on Monday we'll come back and we'll talk about the other kinds of hypersensitivity responses, followed by a lecture about autoimmune disease.